that's called inositol triphosphate, or IP3 for short. And IP3, the inositol triphosphate, is also a second messenger. It's also a small non-protein molecule that's going to drift around inside the cell, diffuse around inside the cell. So where does inositol triphosphate come from? Well, the short answer is from the phospholipids of the membrane. Remember, phospholipids have these areas called the hydrophilic heads. IP3 is one of those hydrophilic heads. So this phospholipase C is essentially lopping the heads off some of the nearby phospholipids. When we were talking about phospholipids earlier in the semester, I showed you this diagram. Phospholipids are always consist of a glycerol, 1, 2, 3 carbons in a row. To 2 of the carbons of the glycerol, you have fatty acids attached, like this one and like that one. Those form the 2 legs of the phospholipids, the hydrophobic tails. To the 3rd carbon of the glycerol, remember there's always a phosphate group. And when I was introducing this concept a few weeks back, I said that on the other side of the phosphate group, in other words over here, you always get some atoms, but exactly what atoms are there kind of differs from one phospholipid to another. Well, there's a type of phospholipid that has a certain hydrophilic head group here called inositol triphosphate. And so what I'm saying is the activated phospholipase C finds that particular phospholipid in the membrane and chops its head off. So you get these hydrophilic head groups, the inositol triphosphates being released, and since they're hydrophilic, they drift away from the membrane and just go out into the cytoplasm. And so they count as a second messenger. I think I've neglected to mention something. So these second messengers are always small non-protein molecules. Why are they small? Well, remember the laboratory on Wednesday, the smaller the molecule, the faster it diffuses. So by being small, second messengers can spread out in the cytoplasm faster. Okay, well, from what I've said so far, you might think, aha, I know what's going to happen. Those inositol triphosphates will be allosteric activators of the next relay protein. But it's not quite that simple. The IP3s do not directly activate the next relay protein in the pathway. To see what they are going to do when they drift down to the cytoplasm, we got to drag the endoplasmic reticulum into the picture. When we were talking about cell structure a few weeks back, I talked about the endoplasmic reticulum being a series of membrane-bound compartments connected to the nucleus. Who remembers what the function of the endoplasmic reticulum was? It was a manufacturing center. Remember, the ribosomes that make the proteins attached to the endoplasmic reticulum. What else gets manufactured inside the endoplasmic reticulum? Lipids, right? It's also a lipid factory. And those are the major purposes of the endoplasmic reticulum. But something I didn't tell you at the time is this. The endoplasmic reticulum also serves as a storehouse for the cell's calcium ions. There's a really high concentration of calcium ions stored up inside the endoplasmic reticulum. And this little protein right here in the membrane of the endoplasmic reticulum is a transport protein, a channel protein that can let those calcium ions diffuse out. And so what IP3 actually does, this second messenger from the phospholipase C, is it binds to that transport protein right there and gets it to open up. And so now, by diffusion, that high concentration of calcium ions will now exit the endoplasmic reticulum and go into the cytoplasm. And the calcium ions act as the second messenger to activate the next relay protein, like you see there. And from this point on, it's kind of a regular signal transduction pathway. That's probably going to be a kinase of some sort, maybe a tyrosine kinase, maybe a serine threonine kinase, but it'll probably add phosphates to the next relay protein, which will probably kinase the next relay protein until we finally get down to the cellular response enzyme at the end of the pathway. Sound good? Are you impressed? Amazing. Cells are amazing things. For something that you so small you can't even see it, there's a lot going on inside there. Well, that's probably a good stopping point. Let's put our stuff away and do the weekly quiz.